dear, dear students. So yesterday we learned regarding the introduction to the nucleic acids. So then, so what is the nucleotides? So their structure, then types of the nucleotides. They are mainly two types: purines and pyrimidines. The purines are adenine, guanine, and pyrimidines are cytosine, thymine, and uracils. How they are contribute in the formations of RNA and as well as DNA regarding the nomenclature, then as well as their sugar. So RNA containing ribose sugar, DNA containing D-deoxyribose sugar, and as well as so why the continuous supply is required regarding the supply of the nucleotide, that is also studied. So because they are involved in the genetic informations or their source of energy, and as well as they are required for the formations of FD of the glucose, that is the glycogen, and as well as the main important function. So they are involved in the signals transductions. So signal transduction, that is the role of the nucleotides. Okay, so this signal transduction is also essential for the growth of the disease also. Okay, microorganisms, so they are multiplied, the disease also growth or multiplied simply. Okay, so how it is possible? There is a signal transductions between the microorganisms, so they are involved in the to enhance the or growth of the disease. If any drugs or any phenomena, if cut that signal transductions in the microorganism means, so that drug is successfully helpful for treatment of the microbial infection also. And also we started regarding the biosynthesis of ribonucleotides, regarding the biosynthesis of adenine and guanine. So these are occurs mainly the two steps or two pathways, that is the de novo synthesis pathway. For the purine synthesis, that was the major pathway. It is utilized the certain components or raw materials, then it was involved in the synthesis of the purines, and it is the major pathway. Then slavage pathway, the byproduct they are utilized. So due to metabolism of purines, certain byproducts are releases are formed, or even the hypogenthines, it is the byproducts of the various purines, so that was utilized for the synthesis of the purines. So that pathway is known as the slavage pathway. It is the minor pathway. And also we learn regarding the, so how the various components are amino acids, so they are contribute in the formations of the basic ring of the purines. So that is N1 donated by the aspartate, then C2 C3 is distributed by that is the formate, then N3 N9 another two nitrogen that was contributed by glutamine, then C4 C5 N7 it is contribution of the glycine, and as well as C6 it is the contributions of the bicarbonate. Okay, that is the carbon dioxides in the lung, it can react with the water, it can forms the bicarbonate. So that bicarbonate was helpful for the to add the carbon 6 atom in the purine ring. So like that, so these are the various components, so those are involved in the or contribute in the formations of the purine rings. And as well as the purines adenine and guanine, so they are synthesized or derived from the inosine 5 monophosphate IMP. So that's why inosine monophosphate, it is one of the main intermediate from which only the adenine and guanine, the purines, so they are synthesized. So now we learn regarding the locations of de novo synthesis of purine, so where it is occurs. So the purine synthesis occurs in all tissues. So mainly it can occurs in the liver and as well as other tissues, but limited but negligible, or it is not happen occurs in the brain. So major site for de novo synthesis of the purine, it is the liver and as well as many other tissues, but negligible or not occurs in the brain only. So regarding the substrate, so that was required for the biosynthesis of purine is a 5 ribose phosphate. So already know 5 ribose 5 phosphate, it was the end products from the pentose pathway or HMP pathway or hexose monophosphate pathway. So they are formed the ribose 5 phosphate, and ribose 5 phosphate is the intermediate compound or precursor for biosynthesis of the purines. Okay, so then as well as substrate required, it is a glycine, glutamine, water, ATP, carbon dioxide, and as well as aspartate. So these are the various substrate. So that was required for the synthesis of the purines. And the products, end products after the biosynthesis of purines, the final products are produced like GMP, that is guanine monophosphate, then AMP, adenine monophosphate, and as well as the glutamate, fumarate, the water is formed during the synthesis of the purines. So regarding the overlook or overview of this pathway or purine pathway, de novo synthesis purine pathway, so it can utilize the ribose 5 phosphate that was supplied by the pentose phosphate pathway. So that was converted into PRPP, that is the phosphoribosyl pyrophosphate by an enzyme PRP synthetase. It is the first step. It was also required certain ATP, and that 5 ribose phosphate that was got from or produced from the pentose phosphate pathway. So these enzymes are in the presence of this PRPP synthesis enzyme. The ATP is phosphorylated. Phosphorylation means the release of phosphate group. So that phosphate group, it is attached to the added to the 5 ribose 5 phosphate, then form the PRP, that is phosphoribosyl that is the pyrophosphate. Then next step, that is regarding the general overview. So that is the commitment steps. Okay, that was the formations of conversions of glutamine, that is the PRP, into a 5-phosphoribosyl amine. So this is catalyzed by the PRPP. Then there is a further 9 series of reactions are involved in the formations of IMP, that is the inosine 5 monophosphate from the ionic, that is the IMP. Then GMP is produced by I, that is the IMP dehydrogenase. Then as well as then AMP is produced by a adenyl succinate synthetase enzyme. So this is just an overlook regarding the de novo synthesis of the purine. So main is the basic molecule is ribose 5 phosphate. Then it was involved in the formations of PRP, and finally it was involved in the, after going the series of reaction, total 9 reactions are there, the IMP will be the forms. So then that IMP is required for the formations of guanosine as well as the formations of the adenines. So now we learn the step by step. The first we learn regarding the synthesis of 5, that is the inosine 5 monophosphate. So inosine monophosphate, so that was found by going through the series of reactions. So beginning the precursor, so that was involved in the precursor that was required for the formations of inosine monophosphate is the ribose 5 phosphate. So this is the first molecule or precursors. It can begins the, it is required for the formations of the inosine 5 monophosphate or IMP. So that's why the first step involved in the de novo synthesis of IMP is, that is the ribose 5 phosphate activation and formations of the PRPP. So here the ATP, so that was phosphorylated. Okay, so that was ATP is eligible for to donate the phosphate group to the alpha D ribose 5 phosphate. So that was catalyzed or regulated by an enzyme. So that enzyme was known as a PRPP synthetase enzyme. So this enzyme, it can cause the additions of, that is the phosphate group. So that was released from the ATP. Then it was responsible for the formation of the 5-phosphoribosyl alpha pyrophosphate. So that ATP, it can use the phosphate group. Then the ATP is converted into AMP. So like that, it can give the 2 phosphate group to the ribose 5 phosphate. Then it becomes the 5 phosphoribosyl alpha pyrophosphate. The second step, that is the acquisitions of nitrogen atom in position 9, ring, are the 9 positions of the purine rings. So here, so this glutamate, it can transfer the amide nitrogen to the PRPP or to the phosphoribosyl pyrophosphate. So that glutamine, it can transfer the amide nitrogen to the PRPP. Then it can catalyze the pyrophosphate from the PRPP. Okay, it is one side. It can give the amido group, and as well as, so this enzyme, that is the amidotransferase, it can remove the pyrophosphate or catalyze the pyrophosphate from the PRPP. Then form the, that is the beta 5, that is the phosphoribosyl amine. So like that, the beta, that is the beta 5 phosphoribosyl amine was formed when the glutamine, it can donate the amide nitrogen, then as well as the amidotransferase enzyme then can cleave the, so this pyrophosphate under the influence of the water. So in this reaction, the water molecule is required, then glutamine, it can the source for the amide nitrogen for the position N9. So like that, that is the beta 5 phosphoribosylamine that was found from the PRPP in the presence of the glutamyl amidotransferase and glutamine, and as well as the additions of the water. So next step, that is step 3, that was the acquisition of C4C5N7 atom of purines. So in the step 3, the carbon atom 4 is added, carbon atom 5 is added, as well as, that is the another nitrogen, so that was added at the 7th positions. So here the synthetase enzyme, so that can convert, that is the beta 5 phosphoribosylamine, okay, into a glycinamide ribosyl phosphate in the presence of the glycine and as well as the ATP. So ATP, it can give the phosphate, okay, in the presence of the synthetase enzyme and glycine enzyme. Then, so this synthetase enzyme can convert or responsible for the formations of the glycinamide ribosyl phosphate. So that is also known as the GAR, that is the GAR. And further, that is step 4. In the step 4, the carbon atom is added at the 8th positions of the purine ring. So that's why it is named as the acquisitions of the C8 atom of purines. Okay, so here, that is the, N10 formyl tetrahydrofolic acid from the formate. So it was donate the formyl group to the glycinamide ribosyl 5 phosphate and forms the formyl glycinamide ribosyl 5 phosphate, okay, in the presence of the enzyme formyl transferase enzyme. So this formyl transferase enzyme, it can convert GAR into a, that is the formyl glycinamide ribosyl 5 phosphate by presence of the formate. So that formate, it can donate the formal group to the GAR or glycinamide ribosyl 5 phosphate. The step 5, that was known as the acquisitions of N3 atom of purine. That is the additions of nitrogen atom in the 3rd positions of the purine ring. So here once again, the glutamine, it can transfer the second amido groups or second amido amino group to the formyl glycinamide ribosyl 5 phosphate in the presence of the synthetase, and the phosphate group donor is, that is the ATP. So in the presence of ATP and glutamine, the synthetase enzyme can convert formyl glycinamide ribosyl 5 phosphate into a formyl glycinamide ribosyl 5 phosphate. So like that, the glycinamide was formed from the 5, that is the formyl glycinamide ribosyl 5 phosphate in the presence of the synthetase enzyme. In the step 6, that was known as the purine imidazole ring formations. So the imidazole ring, so here it is the open. So in the step 5, So that ring was closed. So that's what is known as also known as the closing of the imidazole rings or formations of the imidazole ring. It is also required the ATP. The same synthetase enzyme, it can responsible for the closing of the ring to form say 5 amino imidazole ribosyl 5 phosphate by utilizations of the ATP. So like that, so the ring was closed, so then they were responsible for the formations of the imidazole ring for a purine synthesis. So next, that is the step 7, that is known as the acquisition of carbon atom 6 of purine. So in this step, the carbon atom was further added at the sixth positions. Okay, it is also known as the carboxylations reactions. It is only one step in the biochemical reaction, so where the carbon atom is added, but so no ATP is required. Okay, so here the carbon donor is a carbon dioxide. So it can react with water, it can form the bicarbonate. So that bicarbonate can donate the carbon atom to the 5 amino imidazole ribosyl 5 phosphate. So this reaction was catalyzed by the carboxylase enzyme without any need of the ATP. So this carboxylation is occurs and yield the 5 amino imidazole carboxylate ribosyl 5 phosphate. So that product can be yield after the carboxylations reaction that one regulated by the carboxylase enzyme. The step 8, that is the acquisition of N1 atom of purine. So in this step 8, the another amino groups or amide group that was added to the purine ring. So here the amino group donor is a aspartate. It was also required the ATP. The synthetase enzyme, so that can utilize the aspartate and ATP or utilize the I use the amino group from the aspartate and add to the 5 amino imidazole carboxylate ribosyl 5 phosphate and responsible for the formations of the 4 amino imidazole 4 that is sorry 5 amino imidazole 4 succinyl carboxamide ribosyl 5 phosphate. So like that, that is the in the step 8. So N1, another nitrogen atom, can be contributed by a aspartate amino acid. Then step 9, that is the eliminations of the fumarate. So in the 8th step, the fumarate was added to the purine ring, so that was eliminated in the step 9 in the presence of the adenyl succinate lyase enzyme. So this enzyme, it can eliminate the fumarate from the 5 amino imidazole 4 succinyl carboxamide ribosyl 5 phosphate and responsible for the formation of 5 amino imidazole 4 carboxamide ribosyl 5 phosphate. So like that, the fumarate was removed from the basic rings or from the purine rings. In step 8, that is the additions of the carbon atom at position 2 of the purine rings. So once again, so here the fumarate or N10 formyl tetrahydrofolate. Okay, so that was donated the another carbon atom to the 5 amino imidazole 4 carboxamide ribosyl 5 phosphate in the presence of the formyl transferase enzyme. So then the 4, that is the 5 pharma amino imidazole 4 carboxamide ribosyl 5 phosphate is formed in the step 9, that is step 10. So another carbon atom can be added in the presence of the formyl transferase by utilization from the N10 formyl tetrahydrofolate. Last step, that is the formation or cyclizations of the basic molecule. So that was required for the synthesis of adenine and guanine. That was the IMP, that is inosine monophosphate. So in this step is known as the cyclization to form a IMP. This is the final steps. So this reaction was regulated by cyclohydrolase enzyme. Okay, so this reaction was regulated by the cyclohydrolase enzyme. So this enzyme, it can eliminate the water molecule from the 5 pharma amino imidazole 4 carboxamide ribosyl 5 phosphate and responsible for to form the bond or to form the closure of the ring. Okay, as for formations of the bond between nitrogen and carbon atom, then responsible for the formations of the IMP or inosine monophosphate. So like that, the inosine monophosphate was formed. From the inosine monophosphate, the purine and that is the purine nucleotides like a guanine and adenine will be the forms. So in the next step, we should be learn regarding the synthesis of the AMP, that is adenine. So once the IMP was formed, so that was the basic molecule. So that was required for the further the formations of the AMP, that is the adenosine monophosphate, as well as GMP, guanosine monophosphate. So IMP, so that was formed, AMP that was formed from the IMP by a two step. In the first step, there is a amino group was added to the IMP by a aspartate in the presence of the enzyme. So that was known as the adenyl succinate synthetase enzyme. So this enzyme, it can utilize the amino group that was supplied by the aspartate, then responsible for the formations of the adenyl succinate, and where the GTP can convert into a GDP and one phosphate group can be released. In step 2, once again, the adenyl succinase enzyme, okay, it can remove the fumarate, okay, from the adenyl succinate and responsible for the formations of the adenosine monophosphate, and that is the AMP. So like that, the AMP was formed by a two enzyme from the IMP. One is the adenyl succinate synthetase. It can got the phosphate and as well as amino group from the aspartate. And second, adenyl succinase, it remove the fumarate from the adenyl succinate and responsible for the formations of the adenosine monophosphate. So like that, the GMP, that is the guanosine monophosphate, it is also formed by the two steps, that is the dehydrogenations of IMP as well as the amidations of XMP. So IMP, so that was converted into XMP, that is xanthosine monophosphate. So that was regulated by IMP dehydrogenase and utilize the NAD and water and form the NADH plus and as well as xanthosine monophosphate, that is also known as the XMP. So this was formed from the IMP in the presence of the dehydrogenase enzyme, and further there is GMP synthetase enzyme in the presence of the glutamine, ATP and water. It can convert XMP into guanosine monophosphate, that is the GMP. So like that, the AMP, that is adenosine monophosphate, then GMP guanosine monophosphate, so they are formed from the inosine monophosphate. So like that, the purine rings like AMP and GMP, so they are formed from the IMP by a de novo synthesis. So regarding the important enzymes and regulations of de novo synthesis of purine, that is the PRPP synthetase. So this enzyme required for the regulations of de novo synthesis of purine, and this enzyme was inhibited by AMP. So as the AMP concentration increases, automatically that enzyme activity was decreases. Even IMP and GMP. So these are the end products of the biosynthesis of purine. If they are concentration increases, automatically so they are inhibit the activations of the PRPP synthetase. Next, so they do not involved in the formations of the IMP. So another most in enzyme play important role in the de novo synthesis purine is the glutamine PRPP amidino transferase enzyme, and this enzyme, it was inhibited by a once again AMP, IMP and GMP. So another, that is the IMP dehydrogenase enzyme. So it was inhibited by the only GMP. So another enzyme that is important in the synthesis of purine, that was the adenylyl succinate synthetase, and it is inhibited by the AMP. Then other certain synthetic inhibitors are there, or drugs are there. So they are also inhibit the de novo synthesis of purines, like a mercaptopurine, methotrexate, azathioprine, thioguanine, azaguanine. So these all are the antibiotics used as a anti-cancer drug or treatment of cancer. So those are also inhibit the de novo synthesis of purine, both in the human beings as well in the cancer cells, healthy cells as well as cancer cells. And another, there is sulfonamide, trimethoprim. So these are the synthetic antibiotics. So they are helpful for to inhibit the bacterial infection any kind. So these sulfonamide, trimethoprim, they are also inhibit the de novo synthesis of the purine. So both in the healthy as well as in the microorganisms. So these are the synthetic inhibitors. So they are inhibit the de novo synthesis of purine. But the de novo synthesis of purine is mainly depending upon the enzyme PRPP synthetase, glutamine PRPP aminotransferase, then IMP dehydrogenase, and adenyl succinate synthase. So these are the four enzymes. So those are required for the regulations of the de novo synthesis of purine. But they are also inhibited by AMP, IMP as well as the GMP. So next one. So regarding the that is the purine slivase of pathway, that is the synthesis of the purine from the intermediate compounds, not a that is from the metabolites, not a precursors, not a components, from the intermediate metabolites like a xanthine or PRP. So from where directly, so the certain purines are synthesized, and that pathway was known as purine slivase pathway. So locations regarding the purine slivase pathway, it is occurs in the all type of the tissues, especially in the brain and as well as in the bone marrow. So what are the metabolites are produced after the catabolism of purine? So those are reutilized for the formations of the purine by a brain as well as bone marrow. So regarding the substrate for purine slivase pathway, that is the hypoxanthine as well as PRPP, guanine, and adenosine. So these are the substrate from which the purine can be formed. So there is no need of the AMP directly. There is no need of the ribose sugars. Okay, so they are utilized the directly intermediates compounds from which, so they are involved in the synthesis of the end products like a GMP, AMP as well as the IMP. So like that, the purine slivase pathway also involved in the formations of adenine monophosphate and guanine monophosphate. So now we learn regarding the steps. So the purines are slaved by the two different enzymes. So in the manner, so the first enzyme, so that was known as the, that is the adenine phosphoribosyl transferase, also known as the APRT. So which mediates the AMP formation using the PRPP. Okay, so adenine, so then PRPP intermediate compound. So this enzyme, that is the APRT, it convert AMP in and phosphate. So like that adenosine monophosphate, so that was formed from the intermediate compound directly, that is the PRP, in the presence of the enzyme adenine phosphoribosyl transferase, that is APRT. So this is the slavase pathway for the purine synthesis. And second enzyme, so that is the guanine phosphoribosyl transferase enzyme. So that is also known as the HGPRT. So this enzyme, it can utilize the hypoxanthine as well as PRPP and involved in the formations of the inosine monophosphate that was required for the formations of adenine and guanines. So that is the enzyme, that is the HGPRT. It can utilize the hypoxanthine and PRPP, then involved in the formations of the IMP. And also the same enzyme utilize the guanine and reacts with the PRPP or initiate the reactions between the guanine and PRPP and involved in the formations of the GMP. So like that, the AMP, IMP, then GMP. So they are also formed from the slavase pathway. So these are regarding the Slavish pathway regarding the formations of the purine synthesis. So regarding the important enzymes and regulations, so they are mainly regulated by the HGPRT. So then it is inhibited by IMP and GMP. The another enzyme required for the Slavish pathway, that is the adenine phosphoribosyl transferase, and it is inhibited by the AMP. So regarding the purine catabolisms, so after their functions, so they are catabolized, so or they are metabolized. Okay, so nucleotides all undergo the continual turnover. So nucleotides are converted into nucleosides in the presence of the that is the nucleotidase enzyme. Then further nucleosides are converted into free bases and R1P ribose 1 phosphate by a nucleoside phosphorylase enzyme. So some bases are reused for formations of nucleotides by a Slavage pathway, and other products are degraded and are excreted. So end products of the degradations of the nucleotides or purines are, that is the uric acids. So uric acid is the end products of the purine catabolisms. But only few, so they are reused for synthesis of purines, and remaining, so they are catabolized into uric acid, and uric acid that was excreted through a urine. That is the AMP is converted into AMP, it converted into adenosine in the presence of the nucleotidase enzyme, and further adenosine deaminase utilize the water and release the ammonia and involved in the formations of the inosine. And inosine was converted into hypogentin in the presence of the phosphorylase enzyme, that is PNP. And IMP is also metabolized or catabolized in the presence of the nucleotidase enzyme. So that is the additions of water, it can convert inosine and inosine into the hypoxanthine. And further, the hypoxanthine is subjected to xanthine oxidase enzyme. With additions of water, it can convert into a xanthine. And further xanthine, it was converted to uric acid in the presence of the xanthine oxidase enzyme. So like that, the uric acid is formed from the IMP and as well as the AMP. So, on the other side, the GMP can be catabolized in the presence of the nucleotidase enzyme as well as purine nucleoside phosphorylase enzyme, and converted into guanine by the presence of the phosphate and as well as water. And further, the guanine deaminase enzyme, by additions of water, it convert or it can catabolize guanine into xanthine. And further, the xanthine, it was converted into uric acid by a xanthine oxidase enzyme. So like that, the GMP and as well as AMP and IMP, so they are catabolized into a hypoxanthine in the presence of nucleotidase enzyme, then deaminase enzyme and as well as phosphorylase enzyme. Then hypoxanthine was counted into a uric acid in the presence of the xanthine oxidase and xanthine, once again xanthine oxidase. Finally, that uric acid was excreted. And another side GMP. So with the help of these three enzymes, that is nucleotidase enzyme, that is the purine nucleoside phosphorylase enzyme, and guanine deaminase, it was catabolized into a uric acid. So then finally the uric acid was excreted in the urines. So uric acid is a end products of the purine metabolisms.